I wanted to welcome everybody to the Duke Center on Addiction Behavior Change. I'm Ed Levin, Director of CABC, part of DIBS, and welcome to our seminar series. Before I introduce today's speaker, I just want to look ahead. We have a few more sessions in the spring semester schedule here. Dr. Reggie Kennedy, who actually was a student of mine here at Duke as a post-bac for a couple of years, but he's now associate professor at North Carolina A&T, and he's going to talk actually next week on elucidating key cellular substrates in rodents of heavy drinking, alcohol drinking, and chronic ethanol exposure. And then Emily Reisman from NC State is going to be speaking in April on the 13th on sex differences and drug vulnerability. And then Dr. Nisha Godfredson is going to, from UNC Chapel Hill, is going to be speaking for our final talk of the spring semester on April 20th. And that's co-sponsored by Duke Center on the study of adolescent risk and resilience. So now I'd like to introduce today's speaker, Dr. Byron Jones. He's a professor in the Department of Genetics, Genomics, and Informatics at the University of Tennessee Health Sciences Center in Memphis, Tennessee. He's really one of the pillars of the ethanol community here. World leader in the study of behavioral pharmacology of ethanol and its interaction with other drugs. Byron earned his bachelor's in psychology at Eastern Washington University and master's and PhD at University of Arizona in Tucson. And then, not to take any more time from his talk, I just want to get right to it. And he's going to speak to us today about ethanol alters the pharmacokinetics and pharmacodynamics of ecstasy. Byron?
Thanks for the great introduction, Ed. Which world? Say I'm a world leader. Which one?
Earth, I think that was.
Okay, good. Okay. So I guess I'm going to share my screen. Can you all see that?
Yeah, that's fine.
Okay, good.
If you can put it in presentation mode.
Oh, yes. Here we go, right down there. That better?
That's fine. Yep, that's great.
Okay, good. So I'm going to talk to you today a little bit about some of the work I did when I was invited professor at the University Louis Pasteur in Strasbourg, France. They've changed their name back to University of Strasbourg. It's no longer University of Louis Pasteur, but this is the work that we did from 2004 to, and we just got our last paper out last year. So it's been a kind of continuing project headed by a fellow by the name of Jean-Christophe Cassel. So I'm going to tell you how alcohol affects MDMA. Here are the monikers. I don't know where pingers came from. I can understand ecstasy and Molly and Mandy, but I have no idea where pingers came from. And here is the obligatory chemical structure. Here's our good friend ecstasy right here in the middle. And then its first metabolites up on the top right. And so here's some of the other related designer drugs. So I'm going to talk just a little bit about what MDMA does by itself, then we'll talk about what happens when you add alcohol to the mix. So, in pharmacokinetics, it's readily absorbed. 50 milligram dose will get you 106 nanograms per milliliter. And these are all males, human males. And you can see the dose response. And the metabolism has a half-life of 8 to 9 hours, but the effects start wearing off in about 5 hours. So if you're in a long dance party where mostly MDMA is taken, you're going to probably repeat dosing at least once. Its major enzyme to break it down is CYP2D6, which handles a whole bunch of different drugs, and CYP3A4, which, as you know, is inhibited by grapefruit juice. The pharmacodynamics or desired effects increased energy, pleasure, empathy, poikilothermy. The toxicology would tell us that ecstasy produces hyperthermia, But that's normally in dance parties or raves where the ambient temperature is quite high. And then neurochemically, it has major effects in serotonin and somewhat lesser on dopamine in various parts of the brain. Pathology: acute hyperthermia, dance parties, hyponatremia. People are drinking a lot of water because they're really hot, and of course, their sodium concentrations tank. Muscular tension, bruxism, grinding of teeth when they go to sleep, headache, nausea, tachycardia, hypertension, all of the amphetamine-related problems, and rhabdomyolysis, which is breakdown of muscle tissue, which can be hepatotoxic. Renotoxic. Chronic effects: decreased serotonin neurons, particularly if you go looking at the receptors all disappear. They don't disappear. They go down in concentration, which tells us maybe the neurons are also going down as well. Impaired memory, impaired executive function, impulsive behaviors, paranoia, panic attacks, again, the generally usual amphetamine things, psychosis, depression, Parkinsonism. That led a researcher into a rabbit hole a number of years ago. I'll tell you a little bit about that, not much. And cardiovascular and then hepatic problems. So a history of its use, as far as we know, was synthesized by Merck in 1912, patented in 1913, and Merck put a hold on it as a chemical intermediate, and the patent was for protection, not for anything that we knew that it did. Merck never tested MDMA for behavioral effects. 1976, enter Alexander Shulgin. I have a picture of him here next, aka Dr. Ecstasy, who had interest in phenylethylamines, amphetamines, derivatives, and tryptamines, DMT, psilocybin, mostly for psychotropic effects, introduced MDMA to Leo Zeff, who introduced it in his psychology practice. It was noticed that MDMA had made people more empathetic toward other people, and so they thought it might be good for marital counseling. In the 1980s, MDMA became quite popular in dance parties for its social effects. Southern Methodist University in Dallas became the ecstasy capital of the United States. The initial lore stated that MDMA by itself is sufficient and didn't need to use anything else, especially alcohol. It was discouraged. But in the 1990s, MDMA became to be taken with alcohol, and mostly in Europe, and related emergency medical calls increased, mostly hyperthermia-related events. I'll show you what alcohol does to body temperature. In the 2000s, toxicity debate, controversies, and neurotoxic and lethal effects of MDMA. So George Ricordi at Hopkins thought that MDMA would cause Parkinson's disease. So he got a bunch of monkeys and gave them doses of MDMA, and sure enough, saw some neurodegenerative effects consistent with Parkinson's symptoms. And then people couldn't replicate it very well, and so they went back to check what the chemical was he was using. And it turns out he was using high doses of methamphetamine, not MDMA. And he blamed that on the drug supply group at Research Triangle Park, close to you guys. And of course, Research Triangle Park said, "Hell no, we gave you the real stuff." And mostly when you get drugs from the drug supply group, they should send along a chromatogram of what they're sending along with the standard. So he had to retract, I think, 2 or 3 papers. And so people came to think of MDMA as not neurotoxic, but in fact, it probably is. And I'll tell you a little bit about that as well. So there's a picture of Alexander Sasha Shulgin. I never met him. I've been to Berkeley a few times. He had his laboratory outside of Berkeley, I think in Livingston. And he was a synthetic organic chemist who made all these designer drugs and tested it mostly on himself. He tried to get into trouble with DEA, but DEA did support some of his work, but gave him some grief as well. So he thought that MDMA should be used in therapy, and he became known as Dr. Ecstasy. He passed away in 2014, And there you can see a little bit of his laboratory, which always looked like a big mess, but he was fairly productive. So the dance club ethos of the 1980s was ecstasy was sufficient all by itself, no need to add anything else. By the 1990s, it's changed to increase polydrug use, ethanol, and MDMA. By the way, my older son went to SMU and graduated in the 1980s. And I asked him if he knew anything about ecstasy. He said, "No, Dad, of course not." "Yeah, sure." So ethanol and MDMA use became more pronounced in Europe than in North America, increased emergency room visits for those intoxicated with both. And then there was near total absence of preclinical research on the interaction between the two substances. So moving on to 2004, I was invited to the University of Louis Pasteur in Strasbourg to give some lectures on pharmacology to the doctoral students in psychology and biology. And during that time, I met with one of the researchers, pictured here. His name is Jean-Christophe Cassel, who is a very productive neuroscientist. And he does a lot of work with lesions in hippocampus and cognitive problems, and then how to fix them. And so you can look him up on PubMed. He's very, very proficient. So he asked me if I would help him develop a protocol for looking at the interaction between alcohol and ecstasy. And I said, "Sure, I've done this kind of work before." So, but first we had to do some work with MDMA, see what MDMA did to our animal model. And most of you will know me from my work with inbred strains of mice. I do a lot of neurogenetics, toxicogenetics, and pharmacogenetics. In this case, we did no genetics work at all. So the experimental model we had for MDMA was to figure out the dose response. So our subjects were long Evans rats, both sexes, starting at 49 days, dosing at 49 days of age. The doses we did were 2, 5, and 10 milligrams per kilogram intraperitoneally administered. We mixed the MDMA in the alcohol. We made, that's later. We just did one injection of MDMA, and then we dosed it 3 times at 2-hour intervals. The endpoints were body temperature, because we wanted to see the poikilothermic effects. Activity, we had activity monitors in their home cages, so we could measure back and forth movements and record it automatically. And that was just for the 10 milligram per kilogram dose. The overall goal was to emulate repeated dosing as might be observed in dance parties. So people who go to dance parties will take a loading dose maybe before they go to the dance party, and a few hours later into the dance party, they feel the effects wearing off, so they'll take another dose. And sometimes they'll take as many as 3 doses. So here's the schedule. Here's time 0, 2 hours, and at 4 hours. So here's the effect of MDMA on body temperature. Notice we have some doses down below that are kind of not terribly different from saline. And look what happens when we go from 5 milligrams per kilogram to 10 milligrams per kilogram. The 2 top figures, the males and the females. And the females are the open squares, and the males are the closed squares. And you can see that the males are more affected than the females. Okay? And this is after the second dose. Here's the effect on locomotor activity of crossings back and forth in their home cage. And you can see that the MDMA produces a big effect on locomotor activity. And again, the males are more sensitive than the females. But here's what surprised us and really gave us pause to think what we were doing. We looked at the survivors after the second injection, and 3 of the females died. 7 lived on to be tested for the third dose. But after the second dose, all the males died. And we weren't expecting that at all. No males survived at all. So for the animals, for the female, can you see that? That's pretty. I had my scanner on my printer doesn't work very well anymore. So I tried to take this with my camera on my phone. But you can see over here, this is, we let the females live another 12 days. And on the 13th day, we euthanized them and took out the brain and dissected out the prefrontal cortex, the frontal cortex, the temporal cortex, occipital cortex, dorsal and ventral hippocampus, and we measured catecholamines and serotonin and 5-hydroxyandroacetic acid. So we saw no effect, no lasting effect on the catechols at all. But we sure saw it with the serotonin, and we saw a decrease in the concentration of serotonin and 5-HIAA in all of the brain sections that we looked at. So this told us that there are some lasting effects of exposure to MDMA, the three times exposures, and again this time we couldn't measure the males. The males were dead, but we saw this in the females. Okay, so then moving on, we had then to figure out what we wanted to do about alcohol and MDMA. So the experimental model we developed here was to do a dose response with 6.6 and 10 milligrams per kilogram. The dose that killed the males was 10 milligrams per kilogram, so we backed off. That's why we backed up to 6.6, and we saw actually I don't think we saw any fatalities with 6.6. As again, and we mixed, and the dose of alcohol was 1.5 grams per kilogram. We mixed it up, I think, in 20% by volume, and we combined the two drugs. So the animals just got one injection. Dosing, we didn't do 3 doses here. We did just 1 dose. Sorry, that's a mistake on that slide. So the endpoints again were body temperature taken rectally and activity, back and forth movements in the home cage. So looking at locomotor activity, we saw our initial observations in the hoods were a synergistic effect of alcohol and MDMA-induced hyperlocomotion that showed tolerance across successive days, but not related to tolerance to alcohol. As a matter of fact, in a separate experiment, we put the animals on alcohol for several days before we did this, and we saw the same effect. The dose of MDMA was 10 milligrams per kilogram, and in this case, the ethanol was 1.5 grams per kilogram. In this case, the alcohol seemed to protect against the lethality of the MDMA. And the bar graph up here shows you the effects of MDMA by itself and MDMA plus alcohol. So in terms of body temperature, sorry for the fuzzy on this. Again, this is from my scanner. We saw that on the first day, ethanol tended to protect against the hyperthermic effect of MDMA. It's interesting because both agents are poikilothermic. So we got protection against hyperthermia on the first day of injection, but got tolerance to that on the second day. And again, it's not related to tolerance to alcohol. It's tolerance to the combination of alcohol and MDMA. Okay, so you see the effects over the days, and you see a little bit of tolerance just to MDMA by itself. Okay. So then we wanted to see what happens when you give MDMA and alcohol on a different schedule, like every other day. And so here's a schedule. So we had 3 days of just saline injection and put them in the home cage to adapt them to being injected, being handled and injected. And then on every other day for days 4 and 6, we gave the treatment, which was ethanol plus MDMA, and then back out on 11 and 13th days. And so this is sodium. So this is saline or ethanol or MDMA on days 4 and 6, and ethanol plus MDMA on days 11 and 13. Okay. So here's what happens. With MDMA and ethanol, on the second day of administration of both agents, we get some sensitization. Look at the area. So we don't see anything here with the effects of the first hour, but look at the second and third hours. We're seeing an increase in the area under the curve for locomotor activity. So it tells us that we're getting sensitization, and especially on days 11 and days 13. Tolerance disappears. We see evidence of sensitization. The pre-treatment with the saline also seems to enhance the synergistic effect of ethanol and MDMA plus alcohol-induced hyperlocomotion. Okay, so what's the neuropharmacology? What's happening in the brain? So we're looking at striatal slices from animals that were treated and on the same schedule you saw before. And this work was done by Rolf Jakisch at University of Freiburg in Germany. And we see here on the left, that's the effect of the combination, saline, ethanol, MDMA by itself, and MDMA plus ethanol on serotonin release from slices. Right, and we see not much of an effect, no effect of different. There's no difference between MDMA and MDMA plus ethanol on serotonin release. But look at dopamine. So it looks like the interaction of ethanol plus MDMA is shifting the neuropharmacology from serotonergic to dopaminergic, because we see a big increase in release of dopamine, which also may account for the big increase in locomotor activity. So, so what about the pharmacokinetics? What's happening with the pharmacokinetics? So here's the effect of MDMA and with and without alcohol on delivery to the hippocampus, the cortex, and the striatum. And you can see here that at both doses, 6.6 milligrams per kilogram of MDMA plus 10 milligrams per kilogram, ethanol increases the delivery of MDMA to all parts of the brain, especially striatum. And as we'll see later, especially the ventral striatum. So let's see what's happening here with some other measures. Here's an experiment with 2-deoxyglucose. You know, it's like the California hotel. You can check in, but you can't check out. And here's this work was done by Anne Pereira Vasconcelos, and so she's doing the imaging with 2-deoxyglucose uptake in the sections. And notice that we see that what lights up here you can't see very well. There's an SE anterior commissure here that you see below the striatum, and then down below you can see where the nucleus accumbens lights up. And to show you in better detail, this is done a later time. Here's a 2-deoxyglucose, saline, ethanol, MDMA, and MDMA plus ethanol, and see that the nucleus accumbens, the core and the shell, both light up. So it's telling us that the pharmacology in the brain is shifting from cortical mostly to striatal and ventral striatal. Okay. And here's glucose uptake in the accumbens. You see MDMA and ethanol. Now you see that's baseline. And you add, MDMA plus ethanol, you see a big increase in glucose uptake and utilization, up by 22 to 25%. So what part of dopamine is active here? So here's 6-hydroxydopamine, which is of course dopamine toxicant. And we see when on the right on panel E here, we see that when you give 6-hydroxydopamine together with ethanol plus MDMA, you see a decrease in the hyperlocomotion produced by the combination. And then, to be more specific, when we add a D1 antagonist, we see that one of the likely mechanisms is D1 activation. And so we're actually shutting that down when we give sharing 23,390 to animals that were given both ethanol and MDMA. So what's next? Well, if we're increasing dopamine activity in these animals, what about reward effect? Is there any? So as you probably all know, MDMA, it doesn't have a high reward effect like cocaine or like methamphetamine, but people take it and they like the effects, but they don't take it compulsively. And likewise, in rats, it's hard to get them to self-administer MDMA intravenously. So I didn't have the equipment to do the intravenous administration. So we reverted to the old-fashioned conditioned place preference. And so what's going on here? So it's saline and ethanol and MDMA all give about the same response in the target box. But look what happens when we give ethanol plus MDMA in the target box. The amount of time spent there increases significantly and dramatically. So this tells us at least with conditioned place preference that there's very likely increased reward effect from MDMA and ethanol combination. Okay. So support our authors that ethanol does increase reward effect. And so conclusions, what on earth? So we know that alcohol alters the pharmacodynamics and pharmacokinetics of MDMA. The change in dynamics seems to favor increased dopamine activity, while the means for the increase of MDMA distribution to the brain is not yet known. One possibility may be solvent drag across the blood-brain barrier. We don't know. We haven't done that experiment. The increase in MDMA by ethanol occurs rapidly, and the rapid bioavailability of cocaine, like the rapid bioavailability of cocaine, is cause for concern. Because we showed that after 13 days after the repeated treatments of MDMA, we saw a big decrease in serotonin content in various parts of the brain. Okay, so if we're getting increased activity in striatum, particularly nucleus accumbens, cause for concern, and together with a conditioned place preference, results are consistent with the effect of ethanol increasing reward effect. So are we increasing the risk for neurotoxicology? So MDMA is considered to be a drug that's not compulsively used, yet the addition of alcohol seems to change the potential for compulsive, possible addiction, and possible neurotoxic effects. Okay, so what's next? Okay, we want to, we actually have, I have a colleague here at the University of Tennessee who does IV self-administration. So I'm going to see if I can get him to participate in the IV self-administration of MDMA in animals that have been treated with alcohol. So we need to clarify chronic neuro and other systems toxicity, probably not just the brain, and genetic-based individual differences in effects susceptibility. We don't know. What about teratogenic effects? Again, we don't know this. And so these are areas that I think that need to be explored. So looking at all the people involved, and I'm going to give a pitch for a program. I'll tell you in a minute. So University of Strasbourg is Jean-Christophe Cassel. He is now the head of the CNRS unit. It's a mixed unit between the center, the French National Scientific Center, and the University of Strasbourg. Sami Ben Hamida did a lot of the work, did his doctoral work with us. He's now he's in Canada now. And Julie Koenig did the first work with just the MDMA by itself. And Anne Pereira Vasconcelos is the imager. She does a lot of the imaging work. Christian Kelch is the previous director of the lab. And Helene Jelch did a lot of her PhD work with us as well as Christine Lazarus. Then moving over to the University of Freiburg in Germany, which is 1 hour away by train from Strasbourg. Rolf Jakisch is a pharmacologist of, I think, about my vintage. He's been around a while. And then Celine Regert and Suzanne Rutz and Katharina Ruthmeyer and Anja Berthelmer. These are all PhD students from his laboratory who also worked with us in Strasbourg. So that's what I have. And I hope that I've convinced you that the combination of MDMA plus alcohol is something to be concerned about. I'll take questions. Amir, yes, unmute yourself.
Brian, it's very nice to see you and hope you're doing well.
Thank you.
Great presentation. A long time ago, I think in 1992, we looked at the effect of this compound on alcohol intake in PRATs and the fun-hooded rats.
Yes.
And actually, I remember, I think we used 5 milligram per kilogram IP, and it reduced alcohol intake and alcohol preference in both lines. That was very interesting. My question is, I hope I didn't miss it. Did you measure the alcohol, the blood alcohol level in rats to see the effect?
Yeah, it was about 175 milligrams per deciliter.
So MDMA had any effect on alcohol metabolism?
No.
Didn't. Okay. All right. Thank you.
Sure. Yeah, was that clear?
Yeah, I'm Jeff George. I saw a new patient of mine. I've only met him twice, and he came in the second time after a party off campus at Duke, and had used MDMA and also alcohol, and this was 3 days afterwards, and he was profoundly depressed.
I would expect that, yes.
Is there any reason to assume that depression will continue?
So Rolf actually did some work with some animals that we kept alive for quite a while. I think 2 or 3, more than 3 weeks after the administration, and their serotonin was still way down. So it's I would imagine. Apparently, if serotonin recovers, it's pretty slow. Right, so you might expect you're gonna have a depressed person for a while.
That's not good news, but I'm glad to know.
that would be my guess.
Okay.
But I'm not a clinician.
Well, I do see him again first of next week, and I've asked him to call me if the depression deepens. So it's pretty profound. So this is very helpful. Thank you.
Are you going to prescribe SSRIs?
We will do, yes. Mm-hmm.
Yeah, but probably that might help.
I apologize. My computer died for a minute. I had to log back on. So anyway, other questions, comments? I had another question in terms of the repeated doses. You had these rapidly repeating doses of every couple hours. Have you, or are you aware of any literature in terms of repeated dosing over days, weeks, months? Is there some cumulative toxicity, especially with the serotonergic systems?
From our own work, we would say probably, yes.
Oh, okay. And then
that's why I'm worried about the combination. If it makes people more likely to take MDMA with be treated with alcohol, probably increase the toxicity.
I see. Yeah, and very typically this is taken along with alcohol in the party setting, I would assume. And in terms of interactions with other drugs, have people looked at that in terms of cannabis or caffeine?
Actually, I had a graduate student who wrote a review paper on that, and I can't remember what's in there. But look me up MDMA and Jones, and you'll see the review paper.
Great. Okay, other folks? It's really very interesting in terms of the way people, actually take drugs, and it's often drugs together. And this is important work. Thanks.
Well, that's the rule rather than the exception. So as you know, that the combination of cocaine and alcohol makes something new, coke-ethylene.
Yeah.
But we don't know about that with amphetamine or amphetamine-derived drugs. So that's the other thing to explore. Are we making anything new?
Yeah, well, certainly, people have looked at the stimulant interactions with opiates as speedballs, and that's particularly lethal as well. So, okay, well, thanks a lot. And so I just want to remind people, we're going to have another session next week, on March 23rd. Dr. Reggie Kennedy from North Carolina A&T is also going to be an alcohol talk, and he's going to be looking at brain systems that underlie that with his rodent work. So, well, thanks, Byron, and thanks
You're
all.
welcome. Good to see you, and Amir, really good to see you.